filtration first, like the basics of it. What is this technique? How it can be used in the field of extracellular vesicles. So you can consider this as a general overview of the technique and its potential applications in the field of extracellular vesicles. And then I will also give some examples using our TFF cartridges, because we have been commercializing these cartridges for some years already, and it had already started to find some applications in the academia or even in the industry. So you can here see a short overview of what we will cover during this presentation. And of course, after the presentation, you can feel free to like ask any questions also during the presentation. If you have any doubts, please feel free to interrupt whenever. So let's start with the basics of tangential flow filtration. Tangential flow filtration is an ultrafiltration method. So it is a size-based method, which is also known as cross-flow filtration. It basically filtrates the samples in the perpendicular direction to a laminar flow, as you can also see in the illustration on the right. So whereas the flow direction is from left to right or from right to left, the filtration to bottom. And it is a widely used method in biopharmaceutical or food industries for purification, concentration, and diafiltration. This means that it is not a method invented solely for extracellular vesicles, but our company, as Hansa Biomed Life Sciences, we are one of the earliest companies to integrate this well-established method to extracellular vesicles. And here you can see a more in-depth illustration of this technique. I hope this video illustration works. So as you can also see in this video illustration, this technique or these filters that are available from us can even be used manually, and they are based on filtrating the samples in sub-millimeter diameters fibers made of non-binding materials such as polyethersulfone or polysulfone. And these fibers are basically illustrated as white thin lines in this illustration. So let me turn this video back on. As you can see, once you are filtrating the sample back and forth, the filtrate is coming out from the bottom filtrate ports, and you can collect this waste in a container of choice. And in the process, the target molecules are separated based on their size. And here it is also important to mention that we are here basically separating the sample according to the pore size of the fibers. If these sub-millimeter fibers have 50 nanometer pore size, for instance, the filtrate coming out of these filtrate ports include particles lower than 50 nanometer, and what remains inside the cartridge includes particles larger than 50 nanometer. But this pore size can be 200 nanometer as well, apart from 50 nanometer. And in this case, what you collect from the filtrate port might be your sample. So it is not always correct to assume that what is coming out of the filtrate is a waste. It just depends on the application. And then I would like to also compare this method to dead end filtration, which is much more commonly known, with different pore sizes or with different applications, with different sample volumes. Since tangential flow filtration is also an ultrafiltration method, what makes it different than dead end filtration is basically the tangential flow direction, because this tangential flow, which is tangential to the pores of the filter, creates a cleansing effect on top of pores, and this prevents the most challenging problem in the dead end filtration, which is the cake formation, as you can see on the illustration on the right part. Over time, particles larger than the pore sizes in dead end filtration accumulates on top of these fiber pores, membrane pores, and then these accumulated particles form a phenomenon called as cake, and this cake formation decreases the flow rate, hence the efficiency of the flow. Whereas in the tangential flow filtration, this cleansing effect on top of pores prevents the larger particles to block the pores, and then there is an cake formation, which ensures steady filtration over time. As I mentioned earlier, this technique was not invented solely for the field of extracellular vesicles. Rather than that, during 70s or 80s, it was developed as a method for purifying proteins in cell separation settings. But as it became more established, it was also introduced into other fields such as food sciences and biopharmaceuticals. But it was only around late 2010s that it started to be embraced by EV researchers as well. And it is also worth mentioning that now in MyCell guidelines, tangential flow filtration is also mentioned as an alternative method for EV concentration. This might be important consideration for some researchers because this MySEV guidelines, even though it is not a strict rule book, it is a guideline formed by multiple researchers. So especially for early level researchers, this guideline is important to follow from many different perspectives. And this guideline proposes TFF as an alternative EV concentration method. And the result of this inclusion in MySEV guideline can be seen in this figure basically, because if you consider this exponential growth, it basically creates a sharp increase after 2018, which was the first MySEV guideline book. And then in 2023, basically in the beginning of 2024, a second MySEV guideline was published by International Society of Extracellular Vesicles. So by the recognition of this MySEV guideline, tangential flow filtration became more and more established in the field of extracellular vesicles to be used for isolation or separation of small or large EVs, EV concentration, or even other purposes related to EV research workflows. But apart from the main technical points of tangential flow filtration, how it can be used, and what advantages, features does it bring to EV isolation? So first and foremost, like the most commonly pronounced advantages of tangential flow filtration is, first of all, its reproducibility, because either for research or for industrial purposes, you need to make sure that batch-to-batch variability in your EV isolates should be minimized in order to reproduce the same results either for diagnostic or therapeutic purpose. And since this is a gentle method which doesn't apply high force or pressure on EVs, or which doesn't include any chemical reaction, either covalent or non-covalent, this is a reproducible method. And it even allows scale up because as Hansa Biomet, we have two filter modules, a small module that can process 10 ml to 1 liter, and a large module that can process 100 ml to 60 liters. And between these two modules, the only thing that changes is the length of the fibers, not the diameter, not the pore size. Therefore, you can ensure that you can perform your pilot studies with the small filter module, and then you can switch to the large filter module without any performance deterioration. This allows flexibility, especially for industrial aims, basically, like application case scenarios. And then these filters are also reusable, which is a factor that deteriorates the cost significantly because you can reuse these filters many times depending on the type of the sample. Here, it is also important to note that the reusability is not a certain number, because this is the most common question we receive for the filters. It depends on many factors. The first is the type of the sample. Like for blood constituents, for instance, like plasma or serum, it can be limited to 2 to 3. But for cell conditioned media, it can even go up to 20. But even for a specific type of sample, it is not easy to define a reusability number or number of reusability because imagine that you are working with cell conditioning media, you might be using FPS as a conditioning medium or a chemically defined media, which changes the number of reusability because of the contaminants inside this conditioning medium. But it is useful to keep in mind that they are reusable many times. And further than that, they are also sterile. All of our TFF cartridges come in sterile packaging. Therefore, you can use them under the hood directly. And even if their reusability is broken, you can easily re-sterilize them through gamma or E-beam irradiation. And of course, the most straightforward approach in order to understand what benefits can be gained from tangential flow filtration is to compare it with the gold standard, namely ultracentrifugation. The main problem of ultracentrifugation is that since it applies high shear force on particles, namely extracellular vesicles, it causes EVs to become sticky and to form pellet, and in many times with fractionated membrane, meaning that you isolate your EVs, but it is questionable whether they are still functional. Whereas in tangential flow filtration, since it is performed through a smooth flow of particles, there is no aggregation and there is no damage on EV membranes. In ultracentrifugation, the recovery of EVs is limited to around 40 to 50%, whereas in tangential flow filtration, more than 80% of recovery is possible. Ultracentrifugation is a time-consuming method requiring hours to process only a couple of tens or maybe like hundreds of milliliter, whereas in tangential flow filtration, hundreds of milliliters can be processed on the order of minutes or tens of minutes. And ultracentrifugation is not a method that is suitable for scale up. You can only buy a new ultracentrifuge to be able to process more volumes, which is not cost friendly, let's say. Whereas in tangential flow filtration, scale up is, as I mentioned earlier, possible from 5 to 10 milliliter up to tens of liters. Ultracentrifugation, since it does not have a strict threshold of particles from any physical-chemical characteristics, it comes with contamination of particles, whereas in tangential flow filtration, there is a strict cutoff set by the pore size. This can be 50 nanometer, this can be 200 nanometer, or 5 nanometer, depending on your application. And this sharp cutoff minimizes the contamination. And finally, ultracentrifugation requires expensive lab equipment, whereas tangential flow filters can be used even manually. But if you want automation, they can be automated with a peristaltic pump. And now at Hansa Biomet, we are also building our own automation system that is adapted to all our filters, and that also has an integrated software, including our optimized protocols, optimized workflows. And with all these comparisons, of course, it is good to mention that these advantages are not only pronounceable when it comes to the comparison with the gold standard, because there are many researchers also aware of the fact that the gold standard ultracentrifugation has some shortages and there are alternative methods in the field. So it is also good to focus on the advantages of tangential flow filtration itself. And the most important advantage is its versatility, because since it is a gentle size-based ultrafiltration method, it allows to be used in many different scenarios, such as separation and isolation of small or large EVs, concentration of pre-isolated EV samples, buffer exchange, removal of small contaminants. This can be unbound dye or unbound antibodies, EV depletion from FPS, for instance, since it is a very expensive alternative as a non-contaminant conditioning medium. You can just buy undepleted FPS and deplete at home. And you can even use it for elimination of unwanted proteins such as THP from urine or casein from milk, whatever you are working with. And even further than this versatility, another good advantage is that it allows the gentle nature of the method allows to be combined with orthogonal methods or other purity enhancing methods such as size exclusion chromatography or anion exchange chromatography. And for combinations like this, the advantages brought by tangential flow filtration is first of all concentration of EVs, because these chromatography-based methods generally work with maybe at most a couple of tens of milliliters. Uh, so if you are working with larger sample volumes, you can concentrate, for instance, 1 liter of sample down to a couple of tens of milliliters, and then this concentrated media can be processed with a chromatography method such as size exclusion or anion exchange chromatography. And while doing this concentration, you can also ensure that you are or you can remove small solutes and large debris effectively with TFF in a scalable and efficient way. And as I mentioned earlier, now we are building this system that you can see. This is a real image of our beta version for automation of TFF-based processing. So this device is built to maximize the reproducibility and minimize the effort for EV workflows. This is still under development, but now available as a beta version. And this can basically process from 10 ml to 60 liters of any EV containing sample in an automated way by the help of our optimized protocols. And now, finally, I would like to mention some examples in order for you to like see TFF in action in different workflows. And I chose these examples to represent the versatility of TFF, both in the sense of processable samples, like cell-conditioned media, body fluids, or like different body fluids, and also for the application scenario. So if you are interested in further examples for any of these cases, you can just contact me for further examples. So in the first case, we see a comparison between induced pluripotent mesenchymal stem cells with uh, mesenchymal stromal cells, sorry, human umbilical cord derived mesenchymal stromal cells. And here tangential flow filtration cartridges are used to concentrate EVs collected from both groups, and then the comparison is performed in order to compare the immunomodulatory and regenerative potential of these two groups in vitro models. And through these in vitro models, it was demonstrated by these researchers that induced pluripotent stem cells demonstrate higher pro-regenerative potential through in vitro scratch assays. And then in a second example, now the EV containing source is bacterial cultures, meaning that bacterial OMVs are isolated with our tangential flow filtration cartridges for their potential use as vaccine development or drug delivery platforms. And here in this paper, the aim is to measure their biodistribution, which is a challenging aspect of uh, introducing the drug to the living organism. And the aim is to establish a high efficiency radio labeling method. And through this in vivo studies performed with the EVs isolated with our TFF cartridges, the researchers were able to demonstrate that their established radio labeling method was showing high efficiency. In a third example, now the source of EVs is a plant, namely citrus, which offers an excellent platform for nutraceutical delivery with endemic cargo, including several different micronutrients. But the problem is their cargo, which is inherently unstable and which is affected by storage and preservation conditions. So the purpose of this this article is to come up with an established storage and preservation protocol. And for this purpose, researchers utilize our tangential flow filtration cartridges to isolate EVs from citrus and then to store and preserve in different conditions, such as fresh juice, frozen juice, freeze-dried juice, and so on, and to compare the effects after these conditions. And with this way, they were able to come up with an optimized workflow. In another example, now the starting material is limbal tissue, in which uh, the role of EVs in human limbal niche homeostasis is not very well understood. And in this study, we can even see the combination of tangential flow filtration with size exclusion chromatography, which was one of the combination alternatives that I mentioned earlier in my presentation for further purification of EV isolates. And here we can see that this combination is suitable for downstream analysis because in this study, the downstream analysis is performed through proteomic analysis, which suggests that these small EVs may be regulating keratinocyte development, extracellular matrix organization, and niche regulation within human limbal niche. And finally, in this last example, now our starting material as EV source is bovine milk, which represents an abundant, cost-effective, and EV source, and it demonstrated good potential in chemotherapeutics or bioactive natural compound delivery. However, the problem is their scalable production from bovine milk resource, as it requires protocol optimization. And this study recommends a combination of workflow which starts with enzymatic casein removal using RNet, and then this is followed by tangential flow filtration as a scalable purification and concentration method for EVs. And these were all the examples I would like to present. So in order to summarize what I presented to you, first of all, TFF provides many different advantages to overcome the present challenges in EV isolation. Of course, it is not enough to say that this is the ideal method because we all know that there is no ideal method for all the possible scenarios in EV related workflows, but it overcomes many challenges in the fields, and its versatility allows that it can be utilized in many different scenarios. And especially after the recognition by MySEF guidelines presented by the International Society of Extracellular Vesicles, it is becoming more and more established and recognized in the field of extracellular vesicles. And since field is now moving more towards applications from basic science, and since the advantages of tangential flow filtration is more important in these industrial settings, it would be wise to expect that in the coming years, TFF would be more and more embraced by EV researchers. And here are some references that I mentioned during my presentation. As I mentioned earlier, the longer version of this presentation is available in the website of Egyptian Society for Extracellular Vesicles, so you can just go there and check, or you can always contact me for further questions. And you can check these references if you are interested for further reading. And that was all from me. Thank you for your attention. If you have any questions, I would be happy to answer.
Thank you, Typhon, for this wonderful presentation. If anyone has any questions, please feel free to unmute and ask right away. Yes.
Good afternoon. Many thanks, Saifan, for the very impressive presentation. It was very illustrative and useful. Actually, I have one question regarding the sterility. So, after the first use of the cartridge, how exactly is the best way to re-sterilize it? Is there a feasible way how to do that? Thank you.
First of all, thank you for the question. It depends actually how you use it, because if you are using it under the hood, the sterility is not directly broken. So until the sterility is broken, you can ensure that it is sterile, and even during reuse, you can use it. But for clearing or washing the filter, we have some protocol established, and that is included in the data sheet. Uh, I don't remember the exact protocol by heart, but more or less, you start by washing the particles with PBS, and then you wash the fibers with sodium hydroxide, and you include some abundant molecule water washing in between to remove sodium hydroxide residues from the fibers. And if there is some persistent color in the fibers, which is generally the case for plasma or serum processing, you continue with sodium hypochlorite, and also after that some abundant molecule water, but this is for washing the filter. So in between reuses, you apply this washing procedure, and then you can make sure that you can reuse the filter. But for sterility, if the sterility is broken, you can re-sterilize with ethanol oxide or gamma irradiation, gamma or E-beam irradiation.
Okay, so UV is not advised or is not recommended in this context, right? Because gamma irradiation is not something that's readily available in our hands in the lab. So
U UV is not recommended. I mean, it is generally not enough because these fibers have like two small diameters. We are considering, let's say, 2 to 300 micron diameter fibers here, and the penetration depth of UV rays are not enough to clean such small diameters. Since gamma rays are much more penetrable, they can penetrate much deeper than UV rays. They are recommended for sterilization.
okay, thank you. But as long as we are using the cartridge under the hood, following the protocol in the pamphlet, all the washing steps and so, there are no worries about the sterility as long
Yeah,
as
yeah, exactly. If you are using it in sterile conditions, you don't need to worry about the sterility because they
okay,
already come sterile.
perfect. Okay, thank you so much. Thank you so much.
Thank you as well.
Typhon, we have a question in the chat.
Mhm.
Did you get to see that, or would you like me to read it for you?
Let me see. Okay, so let me go back to this screen. So this is an illustration. We also have a YouTube video demonstrating its manual use. So I can maybe share this after the presentation so you can even see the full workflow. But basically, these filters have some lower lock connections in the end. So it can be connected to any lower lock syringe. So it is not a syringe that is a consumable of the filter. This can be these like regular brown syringes, 20 ml, 15 ml, larger ones, smaller ones, whatever that comes with a lower connection. So you inject your sample within this syringe, you connect this syringe to one end, and then you connect another syringe to the other end. And then you basically, let me turn on this illustration video again. You basically start filtrating back and forth. Or actually, just a second, maybe I can find this YouTube video quickly and then you can understand even better. Yes. Now I found, let me share my screen again. You can see the YouTube video, right?
Yes, the video
Okay.
is visible now. Yes.
Okay. Um, let me mute this. So you can see the real filter here, and you can also see how small it is. So you basically connect this sample containing syringe to one end, and you can also see how it can be connected. It is basically screwed by the nature of this lower lock connection. And then this is filtrated back and forth like this. You can see that there is the filtrate port here. And once you perform this filtration, particles smaller than the pore size come out of these filters, the pores of the filters. We have three different pore size options: 5 nanometer, 200 nanometer, and 50 nanometer. So based on this pore size, for instance, this one here is a video for TFF EVs, and this TFF EVs product has 50 nanometer pore size. This means that the particles coming out of this port includes particles smaller than 50 nanometer. So this is how you use it manually. You don't need to have some peristaltic pump or this automation device that we built. This is more useful when you are processing much larger volumes, because if you are processing a couple of tens of milliliters, you can easily inject it with a syringe. But if you want to process 1 liter, or if you want to process tens of liters, it is not possible to inject it with syringes. So that's where this requirement to automate it uh, kicks in, basically. I hope it is enough. If not, I can further clarify.
Yes, thank you. If you don't mind, I need to ask about the size of the filter pores. If I would like to isolate mesenchymal stromal cell excessive vesicles, what should be the filter size?
Filter size, do you mean the dimensions or the volume size?
No, the pore size.
Pore size. For mesenchymal stem cells, we also have EVs isolated from adipose tissue mesenchymal stem cells in our product portfolio, because we are also commercializing EV reference materials to be used as positive controls and so on. And for our mesenchymal stem cells, we first process them with 200 nanometer pore sizes in order to remove large cell debris and large vesicles. Then we collect the filtrate and process it with 50 nanometer pore size to remove the small contaminants. So by this combination, you can remove both contaminants larger than 200 nanometer and smaller than 50 nanometer. But of course, depending on your application, for instance, if you want to collect small and large EVs from mesenchymal stem cells together, you don't need to separate maybe larger than 200 nanometer. Maybe you can just use TFF EVs with 50 nanometer pore size to remove small soluble proteins. And then what is left is small and large EVs combined together. We do this combination because we commercialize large EVs and small EVs separately. That's why we need to separate them with some threshold.
Okay, what's the usual dose from 100 ml actually
Uh,
condition?
of course, it changes a lot on the biological source, but in general, we obtain from 100 ml, let's say 30 to 40 vials of our products, and each of our products has 10 to power 11 particle per ml concentration. So you can maybe have an idea about the yield this way.
Okay. Thank you.
Any other questions?
Thank you. We can hear you.
Okay.
Thank you as well.
Thank you, Tayfun, for the great presentation. And you know, the Hansa Biomed products are you can get the Hansa Biomed products now through Madi Medical Supplies. We're the official distributors for them in Egypt. And I've left the link for their product portfolio in the chat. And now, after this wonderful introduction to the TFF technology, I would like to invite you to look at how it can be applied in a complete EV research workflow. So I am very honored to welcome Professor Radwa Ali Mahanna, Professor of Medical Physiology at the Alexandria University, the Director of the Medical Research Center and the Executive Manager at CIRMA, and Founder and President of the Egyptian Society for Extracellular Vesicles. Professor Radwa will introduce CIRMA now and the Egyptian Society for Extracellular Vesicles EV service platform and scientific consultation for researchers working with extracellular vesicles.
Thank you so much, Iman, for this introduction. Can you hear me, all of you?
Yes, Dr. Rado.
Okay, thank you. Thank you for the introduction, and thank you, Tayfun, for the very interesting, comprehensive, and valuable, actually, presentation for the TFF as a technology. I would like to show how we translate this in a complete research service pathway at CIRMA. Which is scientifically endorsed with the Egyptian Society of Extracellular Vesicles. So my aim is not to simply to process samples in CIRMA, but to help researchers choose the right EV isolation strategy to characterize the product probably and report the data in a way that can stand scientifically and support publication. Actually, we were very happy as an CIRMA, which is the Center of Excellence for Research in Regenerative Medicine and its application. It's located in the Faculty of Medicine, Alexandria University, and in the collaboration with the endorsement of the Egyptian Society of Extracellular Vesicles, we are working on the EVs research for years now. And through this, we launched this Egyptian Society, and we have like a full service pathway for the research work with the EVs, starting from the EVs isolation from cell culture, supernatant and biological fluids, with following the MyServ aligned characterization for the particles, the markers, the purity, the morphology. And so we have a coordinated pathway with obtaining of the sample, the preparation, the reporting, and the interpretation. Actually, what I want to say that when we first, I'm so proud that we were the first to have the TFF technology for the isolation of the EVs. And as Tayfun said, there is no a standard way for the isolation of the EVs, but yet, and thus we are using different methods for isolation. We started with the ultracentrifugation, ultrafiltration, and they all had the limitations. When we were introduced to the TFF, we found that it overcome many of these limitations, actually. Mainly the poor recovery, the contamination that can occur from the ultracentrifugation, that we find the EV sticky, the need of this ultracentrifugation equipment. All of this were real limitation when we were working with the EVs. We started then to overcome this with the ultrafiltration, which may have solved some of these problems, but still have problems, mainly the plugging of the filters, which is also overcome with the tangential flow, because this does not happen for the flow of the conditioned media or whatever is the source, while isolating the EVs. So actually in CIRMA, we have the TFF as the first users for them here in Egypt, and of course, obtained from the from Hansa Biomed Life Sciences through Maadi Medical Supplies as the official distributors for them here in Egypt. And it really has this advantages which have mentioned by Typhon, like the scalability, the strong process control, the use for large volumes. There's no plugging for the filters, but yet we can use the ultrafiltration sometimes when we do not need such purity in different. It's according to the research or to what is the aim of the research. We choose the method. We can also combine, like Typhon said, with the TFF using the size exclusion chromatography for having more polishing and gentle purification for more EVs. So actually the method selection, we do not promote one method blindly, but we match the method with the sample and the objective. But starting with the TFF, it may be the start and the end, and we just do the TFF, and we have a very good yield, very good size of the distinguishing or having isolating the large from the small size. It's a very successful step to be done with it. We have also the other ways, because some research work does not need that purification, so we use only the ultrafiltration purification, so we use the SEC. So we have this hybrid workflows. We deliver all of this workflows. Deliver the technical workflow. It is strengthened with the Egyptian Society of Extracellular Vesicles for their interpretation. So the complete EV service package endorsed by the Egyptian Society, from the EV isolation to the particle analysis using the zeta sizing or the NTA, to the marker profile with positive and negative EV markers, flow cytometer or immune assay Western blotting, for the morphology with the TEM imaging, and the research report, which is endorsed by the Egyptian Society of Extracellular Vesicles with the limitations and interpretation notes. So going further than this, what we provide here in CIRMA with the Egyptian Society of Extracellular Vesicles is engineering through the donor cells, the loading strategies, and the scientific consultation. So it is not only a process sample. We help researchers to plan the EV workflow from the start till the end. I hope that we can see you here, and we can be in contact with you either in CIRMA, through CIRMA, or the Egyptian Society. And when you reach us for the isolation, characterization, the research collaboration and project support, workshops, training, and technical services, or scientific consultation in the workflow planning. You can find us in the third floor in the academic building, Faculty of Medicine, Alexandria University, and this is the WhatsApp or the phone number. Also, this is the website for the CIRMA and for the Egyptian Society of Extracellular Vesicles, and on social media in Facebook and LinkedIn, where you can contact us for consultation, for running your research, for using the TFF. As we said, we are the first facility to have it for the isolation of EVs. And thank you so much for your time and for being with us in this introductory presentation.
Thank you, Dr. Radwa, for this wonderful presentation. I've left my email address in the chat box in case anyone would like a copy of this presentation. You can email me directly, and I will also be sending out additional information on the TFF and a link to the presentation, to the longer presentation that Typhon did with the Egyptian Society for Extracellular Vesicles via email. Yes, I will send all of that information out by tomorrow, inshallah, via email. And of course, you can always call your account managers directly for any, if you need anything else.
And
So you
also,
can reach any of us through each other, Typhon through me,
yeah,
through Iman, and we're one entity with each other collaborating for this, for your to help you and to do the best work that we can ever have with the EVs research.
yeah, and also I just sent my email address to the chat. So especially for technical inquiries, like regarding your workflows, you can connect with me.
Does anyone have any other questions before we end this? Well, in that case, I would like to thank everyone for attending today. And you have our contact info. So if you need anything, please feel free to reach out to us anytime by email.
Thank you for your time, and looking forward to your further questions, if there is any.
Thank you.
Have a
Thank
nice
you
day.
so much, everyone. You too.
You
Have
too.
a nice
Thank
day.
you. Thank you. Bye bye.
Bye.